Our next speaker would love to talk about glands, and in fact is going to talk about glands. Leonardo de Oliveira is with us from Sao Paulo in Brazil, and he is a researcher at the world-famous Instituto Butantan, probably very well known to most people in the audience here. And Leonardo is probably the first real card-carrying morphologist who gives a talk in the seminar series. And he's especially an expert on reptiles, in particular the oral glands of snakes, which is, of course, one of the things that makes venomous snakes so interesting. So today, Leonardo is going to tell us about oral glands in a group of snakes that are apparently called goo-eating snakes. I have no idea what these animals are, but it sounds cool. So Leonardo, thank you for taking time to be with us today. The digital podium is yours.
Let me share my screen.
Cool.
Okay, can you see my screen?
Perfect. Yep, perfect.
Yeah. Sorry. Yeah, here. So, first of all, I'd like to thank you, Ronald, by invitation. It is a great pleasure to be here. Also, I'd like to say so sorry about my English because I have no practice in my English for the last time. Sorry by any mistake. Today, I'm going to talk about oral glands in snakes, particularly about oral glands in gypsagene snakes, mainly infralabial and venal glands of gypsagene snakes. So snakes have probably the largest diversity of oral glands among the vertebrates. It is very interesting because snakes are known along their evolution to have lost many structures like legs, like skull bones, but they acquired glands. They have a lot of glands in their head, in particular associated with their mouth. For example, we know approximately 8 different types of oral glands in snakes. The oral gland studies, we can see here many of them. The most famous is this one, the venal gland. The studies of oral glands have been focused on the origin and evolution of snakes and biological functions of the secretion, but most of the studies are focused on the venal glands, evidently because these glands produce venom, and we know the most studies have been focused on each. However, we have little information about the morphological diversity and also about the function of the other oral glands. We can see here in this table, for example, that many information about the function of oral glands are only putative. So, venal glands is a kind of dental glands. In the embryonic point of view, venal glands are developed from a dental lamina primordium. It is like a gland, venal gland, that develops from a teeth in the maxilla of the embryos. We know that the embryos of advanced snakes have two maxillary dental lamina, the posterior one here and the anterior one here. The posterior maxillary dental lamina is related to the development of venal glands and also to the fangs, because both of them are closely associated glands, venal glands, and fangs. The anterior maxillary dental lamina is associated to the other teeth that we can find in the maxillary of snakes. So venom glands of snakes are related to the venom production, and they are composed by several cells associated with the maxillary teeth. They occur in advanced snakes, and they are among the most studied vertebrate cephalic glands. On the other hand, we can see here in an embryonic point of view the development of the labial glands. Labial glands develop from a tissue invagination, the epithelial invagination into the mouth, and they are not related to the dental lamina here, as occur here in the dental glands. These glands here, labial glands, can be found in the upper jaw and the lower jaw of snakes, and they are frequently related to the lubrification, and they are composed by mucous cells. They occur in all studied snakes, and evidently they are much less studied than venom glands in snakes. It is interesting because labial glands in snakes are a series of small invagination in the embryo along the upper and lower jaw, the labial area of the upper in the lower jaw. They are like a gland developed for the many small glands. Okay, so dental glands are associated with the evolution of the venom function of snakes. These glands, venom glands, are thought to be evolved only in the Xenophidian snakes, or also advanced snakes. The group of approximately 3,200 species, Xenophidians are the largest group of snakes we have, and they are also associated to the evolution of the venom delivery system of snakes. Venom delivery system of snakes is composed by venom glands, that associated muscles, and specialized maxillary dentition, and also by the venom produced by the venom glands. We have two types of VDS in snakes, the front phalange and the no front phalange. Okay, front phalange evolved probably 3 or 4 times along the Xenophidian group. They develop in Viparidae, Elapide, and Atractaspidide snakes. In Viparidae, the front phalange system is composed by venom glands. Frequently in these snakes, venom glands has a lumen into the gland where the secretion can be stored. They also have long fangs in front of the mouth associated with a short maxillary, and they also have muscles compressing the gland. And for this reason, front phalange snakes are known as high pressure system related to the delivery of the venom in high pressure from the gland. The other front phalange is found in elapid snakes. They also have large venom glands associated with the fangs, the front fangs. They also have muscles associated with the gland, and they also have an accessory gland where the duct go through to acquire the fang, the anterior fang. The other front phalange system is related to the Atractaspidide. Here we can see the same conjunction. There is a gland associated with an anterior fang. We believe that the front phalange system in Atractaspides evolved 2 times, one time in Atractaspis and one time in Humerus In Atractaspis, venom gland is very differentiated because it is very large, like this one here, and venom glands have association with the muscles and also have a lumen into the gland where the secretion can be stored. The other VDS of snakes are named no front vengeance systems. This system can be found in a large diversity of families of cynophidian snakes. We can see here it is clearly a no natural group of snakes. In this system here, no front vengeance system, we have a gland here in the posterior part of the maxillar, like the front vengeance system, but instead, they have a fang here in front of the mouth. They have here in the posterior part of the maxillary. We have a large morphological variation related to the front vengeance system in snakes. For example, we can find variation related to the venom glands. They can be large like this one, small like this one, or we can not have any gland, any venom gland in some species. The same is related to the maxillary teeth. We can see here a very long here phalange. We can see here a small here phalange, and also we can find species that named no here phalange. We can't find any differentiation between the anterior and posterior teeth. So we also have information about the loss, the secondary loss or reduction of the VDS in snakes. As I told you, Cynophidia is a large group, approximately 3,200 species, and we can find here a lot of variation, but the secondary loss or reduction is not known in the front phalange viperid snakes. We don't have information about the secondary loss or reduction of the structures of VDS in this group of snakes. In Elapid, we have some information about the loss, or better, about the reduction of the VDS. It is present, for example, in species that consume fish eggs, like Ipizurus, Brachyrophis, Emidocephalus. They are marine species. And here we can observe reduced venom gland, atrophied muscle associated, and also a reduced front phalange in species that eat fish eggs. Also, we have some information about the election of some toxins in those species here. And the other group, the most diverse group of cynophidian snakes, we have a large morphological variation. We know little things about the loss, the secondary loss or reduction, but we know that approximately 30% to 40% of species have venom glands. For this reason, we can imagine that a lot of species here have lost or reduction, or have lost or have a reduction in their venom delivery system. For example, we have some information about the reduction in some colubrid species that use constriction to prey, to acquire your prey, like Lampropeltis, Pantherophis. We also have some information about the secondary loss or reduction in gypsadine snakes that consume molluscos and annelids. In both of them, then we have some information about the reduction of glands, or also about maxillary teeth. So, who are the gypsadines? Gypsadines constitute a subfamily of the family Gypsadini. They are here. They occur in America, mainly in Central America, where they are a very diverse group. but also they occur in North America and also in South America. It is very interesting group because into this group, we have two conditions. For example, associated with the type of praise and also about the VDS. For example, in this group here that I have named no-go-eater snakes, we have species that that feed on vertebrates, mainly about frogs, lizards, and fish. And these species here have a hair fungent condition related to the venom delivery system. On the other hand, we have the goo-eaters. Goo-eaters is a name proposed in 1993 for Caddo and Green related to the species that feed on invertebrates of the soft and viscous body, like worms, snails, and slugs. It is very interesting because this group here, goo-eaters, currently have approximately 9 genera and around 300 species. For snakes, it is a very large group of snakes. So we had little information about the maxillary teeth of Gypsagenes, and we had virtually no information about their venom fangs. While we have some relationship between the prey and the fangs, we have no idea about the glands. So for this reason, we decided to perform a study about the VDS of Gypsagenes snakes, and also their possible relationship with their diet. In this study, we studied about 1440 species of the subfamily, including goo-eaters and no-goo-eaters, and we have used many different techniques like molecular phylogenetic analysis, dissections, histochemistry, micro-CT, DICE-CT, and our objective was answer the following questions. Is the VDS of goo-eating snakes, goo-eating Gypsagene, vestigial, or was it lost altogether? Is there any correlation between the morphology of the upper jaw and associated glands of Gypsagenes and their specialized diet? So we divided the Gypsagenes in two different groups. We can see here no goo-eaters snakes that have a maxillary teeth differentiated. They are hair fungent, and they eat about vertebrates. And we found is that we found a very high morphological variation, but all analyzed species have differentiated posterior maxillary teeth. They are hair fungent. We can see here all of them have posterior teeth a little bigger than the anterior one. Most of them have the posterior teeth with grooves. The other species have no grooves or they are ungrooves, but all of them that have differentiated posterior maxillary teeth. Also, we can see here that in this species, for example, Leptodera annulata, we can see maxillary here, and there is a diastema separating the posterior teeth and the anterior teeth. Also, we could find a small differentiation between the posterior and anterior maxillary teeth. For example, we find this kind of ridges here in the lateral region of the tip of the tooth, and also in the medial part of the tooth. This condition here is associated with anterior maxillary teeth. On the other hand, in the posterior maxillary teeth, we find ridges only in the anterior and posterior region. Many authors have said that this marker here can be used in the case where the fangs is not so developed, and we can use this marker here to differentiation anterior maxillary teeth and posterior maxillary teeth, because they developed from a different lamina in the embryo, as I said in the beginning of my presentation. Okay, about this group of snakes, no goo-eaters, we have also some information about the venom produced by the glands, and we also have, for example, here, some evidence that species used your fang, your posterior fang, to probably inject venom in their prey. And about oral glands in the no goo-eater snakes, all of them have venom glands. We can see here there is species that they have a small venom gland like this one, and there is a species that have a large one like this one. We also observed here that while labial glands are mucous, venom glands are serous, and venom glands are also associated with the here fangs like this one here. And about the goo-eaters. Goo-eaters have maxillary teeth where we was unable to differentiate anterior from the posterior maxillary teeth. There is no differentiation between them. We can see here, for example, related to that marker that I told you, related to the ridges, all of them have ridges in the lateral and the medial part of the tooth. So in this case, they have only anterior maxillary tooth. And all maxillary teeth in the goo-eating snakes have only lateral and medial ridges. Many species also have these ridges here that are related to the species that feed on slippery prey. It is probably associated with snakes that need to hold their slippery prey. Okay, and about venom glands. Among the goo-eaters, we have no information about venom glands. We was unable to find venom glands in our dissection, and also we are unable to find venom glands in our histology. For example, here we can see a histological section, and there is no differentiation between supralabial glands and venom glands. We can't find ducts connecting a possible venom glands to the posterior maxillary teeth. We don't have any differentiation associated with the presence of venom glands in this group of snakes, the goo-eater snakes. So our work have the following results. We test the monophily of the goo-eaters, and in our work we find that all goo-eaters gypsagene snakes have a single ancestral. They are monophyletic group. About the venom delivery system structures, we found that while snakes that prey on vertebrates have venom glands and a differentiated here fangent here, all of them have, although there is some morphological variation, someone have small glands, the other one have large glands, all of them have venom glands and here fangent. And about the goo-eaters, we didn't find any venom glands, nor any presence of the posterior maxillary teeth. They are no here fangent. So in conclusion, of this study, we observed that VDS extruders are well developed in gypsagines that feed on vertebrates, but they are absent in goo-eating, like this one here. In goo-eating snakes, secondary loss of venom glands may be associated with the dietary change, like this one. All species prey on invertebrates, and none of them have venom delivery systems here. Simultaneous absence of venom glands and hair phalange dentition reveals the close relationship between both structures. So we know that fangs and glands developed in the embryo from the same structures. So we propose in this work here that probably in goo-eating snakes, they have lost the posterior part of the maxillary lamina in their embryos. So we need to test this in a next study about the embryos. But this proposed here is based on the absence of the glands and of the hair phalange. Okay, but what about the infralabial glands? It is very interesting because in our study, we observed that while goo-eaters have no venom glands, have no hair phalange, they have infralabial glands very developed, and most of them are constituted by seral cells. On the other hand, in no goo-eaters, frequently infralabial glands are constituted by mucous cells. We can see here a clear change in complexity. It is like goo-eaters have an increase in the upper jaw system related to the venom delivery system that was completely lost. On the other hand, they have an increase in the lower jaw conjunction related to the presence of the seral cells in the infralabial glands, but also they have infralabial glands very specialized, like this one here in Geophis. In Geophis, species of Geophis have infralabial glands divided in two distinct portions. We can see here the first one. It is a portion constituted mainly by mucous cells, while this portion here, we name it each of EL2, are constituted only for seral cells. Also, in the posterior, in the anterior portion of the infralabial gland in Geophis, they have a very close, they are very close related to the muscles, like we can see here. Here is possible to see in the posterior part of the gland, they have a compressor muscle here, very related to the gland. And also, they have a lumen into the gland, into the infralabial gland. It is possible the single information in snakes where we observe a lumen into into an infralabial gland, because lumen is only associated with the venom glands, but in Geophis, they have an evident lumen. So the system, the increase of the lower jaw system in Geophis is related to the presence of the two distinct infralabial glands. The first one is related to the muscle. It has a large duct open in the anterior part of the mouth, and they have also a posterior portion of the infralabial gland also associated with the muscle and with the presence of a lumen into each. But about the function of this a very, very differentiated system here. I forgot, is Geophis feed on worms. And in that time that we published this work, we thought about the possible functions and of this infralabial system here. But in that time, we thought that the prey transport and the mucus control could be the better hypothesis to explain the presence of a so differentiated infralabial glands in this genus of species. Because it is not so easy to think about a species need to produce venom to prey a earthworm. But on the other hand, probably they produce mucus, and the control of the mucus they produce, the worms produce, could be important to snakes. So we don't have any information about the feeding behavior of this species, and also about the secretion infralabial gland produce. These species here are found mainly in the Central America. We have also the other infralabial system related to the genus Gypsas, which is here. In Gypsas, we have a different condition from that I told you about Geophis. In Gypsas, we have also infralabial gland constituted mainly by seral cells. We have also infralabial glands divided in two distinct portions, the large one here, EL2, and the small one here, EL1. And a single duct opening anteriorly in the floor of the mouth. We can see here, Gypsas is a genus where species feed on snails and slugs. And the infralabial gland have this totally different architecture. A typical infralabial gland have many small little ducts here, but this portion of the infralabial gland have a large duct here. We can see here the duct in the anterior part of the mouth. But what we can talk about the function of this infralabial system in Gypsas. So before I will show you, we realize we performed a work with the embryos of this species of the Gypsas, and we observed that EL2, that large infralabial gland, invaginate before the other labial glands along the embryonic development, and they have only a single duct open in the anterior part of the mouth, like this one here. And also, the infralabial system of Gypsas is composed by a small gland here and a larger gland here that have only one single duct. The single duct goes to the posterior part of the gland and open in the anterior part of the floor of the mouth. We can see here both parts of the gland, EL1, the little one, and EL2, the bigger one, okay? So, and about the possible function, we know we have some information about the feeding behavior of snakes of the genus Gypsas. For example, we can see here, it is a Gypsas albifrons eating a snake. And we can see in this video that snakes use only mandibula to extract the molluscle from its shell. They don't use the upper jaw system. Like this one, we can see here, it has a lot of mandibular movements, while the upper jaw only holds the shell. And the species remove the molluscle from their shells without break the shells. They use only the mandible to remove the molluscle into their shells. Okay, so probably those snakes only use the mandibular glands to help or not, we don't know, remove the molluscle from their shells. When the gypsies eat on the slugs, they also use only the mandible, and the maxilla is used only to hold the slugs. And the most important part of the to remove to prey the slug is associated with the lower jaw, and for this reason, this system here makes some sense in the feeding behavior of this species. So it is very interesting because we are here studying gypsagid snakes in the America, but we also started to study a group of Asian goo-eater snakes, the Paraid family. And each Paraid family Paraids have a very similar feeding behavior with the gypsagids. And also, when we look for their glands, we also observe a condition very similar. They don't have venom glands here, but they have infralabial glands divided in two distinct portions, like in gypsies. Here, we can see here in these dissections of the EL2 here of parias, we can see the infralabial gland here, and the muscle completely involved this gland. And this condition is very similar to this condition here that we have found in the gypsagid snakes. And also, it is very interesting. Here we can see the muscles, the gland, and about the ducts. Paraid snakes have no only one single duct here, but they have many of them very closely associated to the dentary teeth. So this work is in progress, but it is very interesting because this work here show us a very possible correlation between Asia paraids and American paraids. So, but what about the function of the infralabial glands? We unfortunately have no much information about, but we have information that some authors study the effect of the extract of oral glands in the 19teens, infralabial, supralabial, and venal. They performed some biotests with molluscles, and they observed proteolytic activity of the glandular extract from the infralabial glands. They also observed that the extract from the EL causes 100% of the mortality with injections of the extract. So we have only this information from the 19teens, but more recently, we start to work with the secretion of this species, and we tried two different strategies related to SDS page. We used the extract of the gland, and also we use a gland explange secretion. We did a culture gland expression, and we obtain a very better SDS page, and we use this gland explange to do some biotests. For example, we observed here the proteolytic activity in gelatin zymography and casein zymography related to the infralabial glands of gypsies. Also, we performed some immunoblotting using anti-P3B metalloproteases, SVMP, from venomous snakes, from Bothropis, And we also find some evidence of SVMP in both species of Gypsas, Gypsas indica and Gypsas mecani. We can see here a clear evidence related to the SVMP in infralabial glands. Also, we did some intravital microscopy of the infralabial glands, and we observed using secretion, using the gland explant secretion. We observed some adherent leukocytes, and also we observed a hemorrhagic process in the mycromaster muscles. So we also started to performing some transcriptomic analysis from the infralabial glands, and we can see here is that they have, we can find some typical toxins in the infralabial glands. For example, CTL, crispis, SVMP, interfingers toxins. We find a very a strong intra and interspecific variation, but all of them have some expression of toxin genes. For example, here we can see the SVMP. And also we have found another molecule named eparanases. Eparanases is also being expressed in most of the secretion of the mode of transcriptomes, of the infralabial glands, and eparanases is a endoglycosidase that cleaves heparina sulfate, and heparina sulfate is related to a molecule produced by the slugs and is associated with the mucus production of these slugs, and probably eparanases is associated with the protection of snakes against the mucus produced by the molluscus. So, summarizing, snakes have a great diversity of oral glands, and despite recent advances in the study of oral glands and their secretions, morphological diversity of these glands and their functions remains poorly known for most snakes families. In neotropical goo-eating snakes, the secondary loss of venal glands may be associated with the diet changes, and the simultaneous absence of the venal glands and here phalangeal dentition reveal the close relationship between both structures. Also, the infralabial gland of goo-eating, particularly in the genera Gypsas and Geoffis, is highly differentiated, suggestion additional functions for these glands. Also, infralabial glands of Gypsas species are no homologous tissues. They are not venom glands, express some low amounts of typical venom components, for example, crispis, C-types, lectins, and SVMPs. But also, they are producing molecules probably diet-related. For example, eparanase for dealing with mucus, because mucus is a problem to snakes eat the slugs like this one here. After they ingest these molluscus, they have this condition here. So next steps, secretion is continuing with the work on secretions of the infralabial glands, Gypsas behavior, the morphological oral glands and associated structures in Parieide, the Asian goo-eating snakes. Thank you so much for the audience. So sorry because I think I ultrapassed my time. I would like to thank you particularly Inácio Junqueira for helping me with the studies about oral gland secretions, and also to all collaborators in the study about oral glands of goo-eating snakes. Thank you so much.
Thank you, Leonardo. Very interesting. Don't worry that you went over time. It's okay. That's fascinating. So the question that I have then is, the infralabial glands seem to function as a venom gland in the goo-eating snakes because it kills if you take extracts, it kills molluscan prey. In the non-goo-eating, or maybe you said it, in the non-goo-eating dipsodines, do the infralabial glands also produce these toxins or only in the goo-eating ones?
We have no information about the other ones,
okay.
About the no-goo-eaters snakes, but they have mucous glands, while goo-eaters have serous glands. Also, our start to investigating goo-eatings because they have a totally different gland.
Mm-hmm.
For example, they have glands divided in two distinct portions, but I'm not sure if we have collected any no-goo-eater species to produce, for example, a transcriptomic analysis.
Mm-hmm.
Yeah.
So do you think it is possible that in the non-goo-eaters, or even more broadly, because you said infralabial glands are found in all studied snakes, that they may contribute to venom production in other venomous lineages as well?
Yeah, could be. But it is interesting because they are infralabial glands. They are not associated with the dentary teeth. They are in our idea. It makes more sense related to the feeding behavior expressed by species that eat on slugs and snails.
So when you say the non-gooey eaters, they eat vertebrate prey, amphibians, reptiles, do really none of them eat worms or an occasional snail? I'm just wondering what the reason could have been for this transition in diet.
Yeah, I think we also have no information about the non-eaters eating on invertebrates, or probably if they eat on invertebrates, it is probably a little portion of their diet. What we know about the no-goo-eaters gypsogene snakes is that they feed on vertebrates. We have some species that eat on frog's egg, but no on slugs, worms, or snails.
Also not the juvenile snakes when they're smaller?
I think we don't have this information, Ronald. I think.
Interesting.
Yeah.
Well, you have many more interesting questions in this research program to continue this.
So sorry.
You have a lot of interesting questions for your research program still to work on. So it's
okay. Thank you so much.
nice.
Today I'm very interested about the parage snakes because they are very similar with the American goo-eatings. This is my first question to be solved. I hope.
Very interesting. I see Balint says, great talk. Thank you.